So hello everybody, and thank you for the short introduction. It's called Aarhus in Danish, which is a vocabulary that is quite difficult to pronounce for someone not speaking Danish natively. So my name is Elisabeth Bendstrup. I'm from the Aarhus University Hospital in Denmark, and I'm the leader of the Center for Rare Lung Diseases, and I'm really happy to be invited today to talk about a clinical trial. And I'm going to talk to you about inhaled molgramostim for autoimmune pulmonary alveolar proteinosis, which is a phase 3 trial. And I guess not many of you are pulmonologists, so I hope you can follow me anyhow. So these are my conflicts of interests. And the patients that I'm talking about can look like this on a chest CT scan. So you can see there's a lot of gray-white thing on this chest CT scan, what we call ground glass opacities, and this is caused by accumulation of proteinaceous material in the lungs. This is a somewhat old disease, but we only discovered some of the reasons behind it 25 years ago. The first treatment option was invented in the early 60s, and it was whole lung lavage, where patients in general anesthesia have their lungs washed or flushed with repeated amounts of saline. And it can be quite successful, and then it looks like this. But imagine you have to undergo general anesthesia for 3, 4, 5 hours to have your lungs washed, some patients every second week. It can be quite tough and also a burden for patients. So what is pulmonary alveolar proteinosis? So it's not a distinct disease, it's a rare syndrome. It is characterized by the progressive accumulation of alveolar surfactant. And if you do electron microscopy, as you can see to the lower right, you can see foamy lipid-laden vacuoles in the alveolar macrophages. And from a patient perspective, the disease results in progressive breathlessness, sometimes cough, and also recurrent infections. And for a physician, you will see that patients will have low lung volumes, what we call restrictive lung function impairment, hypoxia resulting in respiratory insufficiency, and in severe cases also respiratory failure, need for lung transplantation maybe, or even early death. So the most common form of the proteinosis is the autoimmune form, where patients develop autoantibodies against GM-CSF, granulocyte macrophage colony-forming factor, which is important for the homeostasis in the alveolar compartment, because the macrophages help to maintain the thin layer of surfactant. If they don't work sufficiently well, if they don't mature, we get the lipid-laden macrophages, and the surfactant will sort of accumulate in alveoli and thus compromise oxygen transport. It's a rare disease with approximately 6 to 27 patients per million inhabitants, and that will correspond here in Ireland to about 30 to 135 patients. And I think, well, in Denmark, where we are the center, we have 16 PAP patients for the last 20 years. So these are indeed rare diseases. Nevertheless, research has been ongoing for more than 25 years, and this is just a summary of some of the studies that has been performed in this rare disease. And just a quick overview, I think you can see the majority have been observational studies. There are only 3 randomized controlled trials published so far. And all the studies differ very much in number of patients, treatment duration, also different dosages. But nevertheless, Professor Cormac McCarthy and me and others thought that it was time to do a European guideline. So the ERS guideline with recommendations for diagnosis and treatment of PAP was published last year, where we did a systematic review and meta-analysis. And here I'm just showing you a few of the outcomes, like the arterial alveolar gradient, the AA gradient, and also the diffusing capacity. Both are measures for how well your lungs can transport oxygen from the alveolar compartment into blood. And both these measures were in favor of treating patients with GM-CSF substitution therapy. And this was also supported by the observational study, resulting in a recommendation where we said we recommend inhaled GM-CSF for symptomatic patients with confirmed autoimmune PAP. This was a strong recommendation, even though the level of evidence was very low. So this is one of the studies that was published in 2020, the PAGE trial investigating GM-CSF called sagromostim in a 1-to-1 randomization ratio with trial arms where patients were randomized either to placebo or to 2 daily inhalation of GM-CSF a week with a week's break in between. So all in all, a total of 12 2-week cycles. And they went on for 24 weeks. And you can see the primary outcome was the AA gradient, And if you have a decline in the AA gradient, it's an improvement for patients. So it met its actual primary outcome here with improvement in the AA gradient. Now, the same year, the first IMPALA trial was published also. It was a phase 2 trial where patients were randomized to molgramostim inhalation solution, which is a form of recombinant GM-CSF, was administered by a nebulizer system called a eFlow nebulizer system. And again, patients were randomized 1:1:1 into 3 groups, placebo or intermittent molgramostim inhalation or continuous molgramostim inhalation. And you can see the primary efficacy endpoint was at week 24. And nicely, the study reached its primary endpoint, which was the AA gradient. There were also secondary endpoints. So this you can see on the figure here on the left. Again, we saw in the study that the AA gradient decreased in favor of molgramostim therapy, and this was also followed by the secondary endpoints that also went in the same direction, so improved diffusion capacity for the lung, improved exercise capacity by a 6-minute walk test, and also improved quality of life. So these very good results fueled the phase 3 trial, IMPALA 2 trial, which has not been published yet, but there was a press release on the company's homepage last autumn showing that the enrollment was finished. Unfortunately, I'm allowed to show you some of the data today, although they haven't been published yet. So the IMPALA 2 trial was again a double-blind randomized controlled trials where patients were randomized to either placebo or continuous daily dosing of inhaled molgramostim, again with a primary efficacy analysis at week 24, but also analysis at week 48. And here the primary outcome was not the AA gradient, which for clinicians is thought to be quite difficult. It needs arterial blood gases and some calculations, whereas diffusion capacity is readily available upon pulmonary function tests. So this was the primary outcome here. There were also secondary endpoints like change from baseline in diffusion capacity until week 48, change in quality of life total St. George's score, but also activity score, and changes in exercise capacity in this study by a treadmill and not by a 6-minute walk test. And there were also exploratory endpoints looking at the burden of protein accumulation by ground glass extent on HRCT scans and also rescue whole lung lavage procedures. So at baseline, the 2 patient groups were very well balanced. And if you look at the 3 key outcome measures like diffusion capacity, quality of life, and exercise capacity, patients were quite similar in between the 2 groups. And also with respect to disease severity, the disease severity score takes into account patient symptoms and oxygenation. And also here patients were quite well balanced. So the primary endpoint was positive, as you can see here. The dark line is the molgramostim-treated patients, and the orange line is the placebo-treated patients. And it's obvious to see that molgramostim was superior to placebo on the change from baseline to week 24 with respect to diffusion capacity of the lungs, but also the benefits were sustained at week 48. If you continue to the secondary outcome measures, first of all, the overall total health-related quality of life by St. George's Respiratory Questionnaire, which is a questionnaire developed for COPD and validated in COPD, but used in many other different respiratory diseases. You see a decline in the score is an improvement in quality of life. So again, we saw what we really wanted to see, that treatment with molgramostim improved quality of life with respect to St. George's Questionnaire total. Both at week 24, it was sustained at week 48. Similarly, for the St. George's activity score, decline is a good thing. So we saw benefit, also statistically significant at week 24, which was sustained at week 48. Continuing to exercise capacity by the treadmill, it was used the peak exercise capacity by METS. Patients on molgramostim increased their exercise capacity significantly at week 48 compared to the placebo group 2. Going on to the exploratory endpoints was made by looking at HRCT scans before and after week 24 to look at the extent of ground glass, which is a proxy for protein accumulation. I think it's very clear from the images on the right to see improvement with less ground glass opacities, And if you look at the score, it was also statistically significant with a larger decline in the treated group compared to placebo. Finally, the rescue whole lung lavage was allowed in both groups. If patient deteriorated, it was at the discretion of the treating physician. I think again it's quite clear here to see that patients in the placebo group needed much more rescue whole lung lavage procedures compared to those on molgramostim. Well, the next question is, of course, is it safe to give patients inhaled molgramostim down to the lungs? And of course, in a study like this, you always look also at adverse events and safety. And overall, there were the same numbers of adverse events, both all adverse events and severe adverse events between the two groups. The number of treatment-related adverse events was similar. Fortunately, there were no death at all. And quite strikingly, very few patients discontinued the drug. If you look at the most common adverse events, they were also quite nicely balanced between the two groups, except for infection with COVID-19 and also for diarrhea. But overall, molgramostim was very well tolerated, and I think that the number 98% of patients on molgramostim stayed on the treatment throughout the whole treatment period really emphasizes that this is well tolerated. I think this number is outstanding compared to many other clinical trials. So, in conclusion, I think that the IMPALA 2 phase 3 trials showed clear positive results with respect to the primary endpoint, which was improvement in DL0 from baseline to week 24. It showed clear improvement in the secondary outcomes, which was DL0 improvement from baseline to week 48, quality of life from baseline to week 48, both with respect to total score and activity score, and also improvement in exercise capacity. And it was well tolerated with a very low treatment discontinuation rate. So I think this study clearly shows that if you have your network in place, because 43 sites was involved in this study, if you have your network in place, it is possible to do very nicely designed prospective randomized clinical trials in patients with rare disease and show benefits of new drugs. Thank you very much for your attention.